I guess just by way of introduction, for those of you I haven't had the pleasure to meet, and there's not many, which is the sign of a good conference. I'm Michael. I'm a PhD student from Southwest Australia, working at Curtin University with the Centre for Mine Site Restoration. I've got a real kind of passion around my project, and it's not a good one to have. I've really kind of positioned myself around difficult to germinate flora and hitting my head against the wall, trying to move some of these things that previously people haven't been able to move. I find this is a really important industry, a really important part of the industry that is kind of underlooked at because it is difficult and that I think really does need to be looked at. We've heard previously about complications with sourcing from wild populations and the ability of industry to meet our current supply, or current demand. And I feel that at least one way we can do that is by increasing diversity in restoration by looking at the proportion of families that we ignore because they are difficult. So in Southwestern Australia, this is how we understand dormancy and how seeds move through soil. So when I'm talking about dormancy here, I'm meaning physiological dormancy. So not physical dormancy that requires permeability and the uptake of water, such as in many acacias and whatnot. I'm talking about true physiological dormancy, which is maintained by concentrations of phytohormones, primarily gibberellic acid and abscisic acid. So seeds in Southwest Australia typically are dispersed in the summer months. They undergo a period of dry after ripening in soil before you start to get autumn rainfall, thunderstorms, showers, what have you. Temperatures begin to drop, soil moisture begins to increase, and seeds begin to go under cold stratification. And this is really your germination window here. Seeds need to establish quickly in these wet, cool months if they've got any hope of surviving the dry periods. In Western Australia, we're lucky enough to have quite a good seed biology history. However, a lot of the research has really focused in germination stimulants. Smoke, being the landmark discovery in the 90s, had a gold rush style application to many, many species within the state. And this resulted in a big increase in flora available to restoration, about 15 to 20%, depending who you talk to. The nurserymen might have found that it's a lot higher, but this is what the literature seems to depict. I believe that if we begin to look at some of these other things in a bit more detail, and some people have with great success, and if we apply them broadly, I think quite easily, we'll be able to start ticking over more species into restoration. Here in my group that I've chosen to work with, the Rutaceae. Now, the Rutaceae occur globally. They occur across Australia. In Western Australia, they are primarily shrubs, very pretty shrubs, I think entirely shrubs. They are important economically. The Rutaceae is the same group that citrus sits within. But in Australia, Borronia megastigmus, which this graph depicts here, is used for essential oil. The areas in red show the current growing area. The areas in yellow show the potential growing area. This industry is worth about $800,000 annually, about 30 hectares of plantations producing about 60 tons of flowers. That industry is constrained by low germination and primarily plants are produced by cuttings. However, we're not overly interested in the commercial viability of Borronia or the Rutaceae as a whole. I'm interested in them because they form a dominant component of species, understory species in Western Australia. So this map, and you can zoom in and these graphs become less convoluted, is something I put together to help inform my collection efforts and where I'm going to source some of these things early on in the project. However, now I'm primarily sourcing seed from suppliers because it is a lot easier and more reliable. They have small seeds, but big enough to work with. They don't contain any obvious covering materials, such as that in the acacia or whatnot, that would make them difficult to germinate. This is true deep physiological dormancy that needs to respond to conditions of temperature and moisture in order to be alleviated and germinate in those winter months. They have developed linear embryos, so no morphological dormancy that's going to slow things down, just true physiological dormancy. So the first thing I did was really go into the lab and try a broad range of treatment applications, and we're talking a lot. Today I'm going to be presenting some of the stuff that has worked and some of the stuff that hasn't worked, but I'm leaving out a lot. So the first thing we really did, this graph shows 5 broad temperature regimes. So we've got 10, 20, and 30 degrees just constant in an incubator. This ran for 14 weeks. We've got seeds that were kept at either 10 or 30 degrees Celsius, and after 6 weeks, we moved them to 20, simulating that change in conditions when the soils are wet. We also exposed seeds to water and carrageenolide. Carrageenolide being the chemical in smoke that makes things go, that stimulates germination. What we find is that these things are difficult to germinate. That is very evident. However, some things do respond to warm stratification quite well. There's no response to carotenoid, which is very interesting because these species are intimately linked to fire, and we know that from their ecology. When we expose them to gibberellic acid, however, the hormone in smoke that's known to bypass dormancy, we get great germination. However, this isn't necessarily applicable for industry. To get this result, seeds had to be plated out for 14 weeks. Sometimes germination took up to 9 weeks. Very high concentrations of gibberellic acid, very long lead times, somewhat costly in terms of time and labor. So while this is important for conservation efforts and potentially for nurseries, it doesn't really give us the whole picture that we need to get these species into restoration and into horticulture efficiently. So focusing in for a moment on these results here in warm stratification, I've just picked out one species in particular. When you increase the time period of warm stratification, so the time at 30 degrees Celsius, what we see from 0 to 1 and 2 weeks is one, an increase in the proportion of germination, but also a widening of conditions that species are able to grow at. So without warm stratification, we don't get any germination at this 5 or 10 degrees. With it, we get quite significant increases in the breadth of conditions that seeds can operate over. This is what we call type 2 physiological dormancy. I'm pretty sure this might be the first time it's actually been confirmed in Southwest Australia, but it is the most common dormancy type globally, so that's not by any means an impressive discovery. However, what this allows us to do is not only increase the number of seeds that we know are going to germinate, but also the breadth of conditions that they will germinate over, which is going to lead to more uniform germination in the field or in the glasshouse, and at the end of the day, produce a better result. And as we move from 2 to 4 weeks, we see a reduction. Where's my laser gone? We start to see a reduction in the germination proportion, and this experiment is currently still running 8-12 weeks, and the broader Rutaceae are undergoing this with smoke applications as well, but because of limited seed, I had to leave it out of this example. So we still got a lot of problems. A lot of these species won't germinate. Mother Nature is the best teacher, so we take the field experiments in various ways. On the left here, we've got seeds sown into punnets and left in open compound conditions to get rainfall. In May, we turned on the sprinklers at the break of the season. Here we've got burial experiments and data loggers, and I'll talk to that after I talk to this. So what we've done here, fresh seeds collected within 4 weeks, treated with either heat in an oven, 90 degrees for 10 minutes, aerosol smoke produced with a bee smoker, or a combination of both. The first thing we notice is there is a response to smoke in the species that we have been getting germination with. So potentially they're avoiding carotenoid, or potentially something confounding has happened here, but I suspect they may be responding to something else in smoke. Most importantly, we see an increase in germination, however small, in species that have been sown into punnets. What this tells us is that nature is doing something that we can't replicate in the lab. Also very significant is this drop in germination that we see for species that we can germinate. So a decrease is likely driven by a loss of viability as seeds weather in summer, or an inability of seeds to emerge. And we know that seed emergence is a confounding factor in restoration success. Here, to kind of investigate that a little bit more, we've got seeds that were soil stored or sown in summer, in January, when they would naturally disperse after treatment. And we've got seeds here that were after ripened in the lab and at the break of the season sown out into punnets. We see two really key things here. One, an increase in germination, likely driven by greater viability as seeds haven't had to endure that harsh summer. We also see that there is germination here. And that's really critical because it means seeds kept at dry, warm conditions have lost a degree of dormancy. Either they don't require summer rainfall events, or rainfall events from May onwards have done something, potentially warm stratification following that break of the season. That's just a close-up of that graph. The difference in germination can be explained by the viability decline of these species. So here we've buried a whole lot of seeds and retrieved them at monthly intervals. And we've done seed viability testing on them. And we've used DRC packages to calculate the decline in viability over time with the model. What we see that in the first 6 months, the viability decrease is somewhat equivalent to the increase that we see here. So that helps to explain that. We see that these things don't hang around in soil for as long as I really expected for at least some things. Certain species hang around for a long time, potentially indicating longer soil longevity and deeper classes of dormancy. Yeah. Here's some pretty photos. So these are scanning electron microscopy images. So we've got species down the left, and we've got time. So fresh seed, 1 month in soil, 3 months, 6 months. So we begin to see a degradation of seed coat. In some cases, we see an increase in microbial activity, and in some cases, we see little to no change at all. So where does this really leave us? We've done a lot of stuff, and we're getting a lot of hints. Maybe this works, maybe that works. We need something kind of stupid in my mind, or stupidly big in my mind, to really delve into this and confirm it properly. So this is an experiment that I put together that I've been running over the past, well, since January. So seeds were either stored in cold storage at Kings Park, just standard storage conditions. They were sewn into mesh bags and buried following treatment in summer. They were hermetically sealed into mesh bags at a fixed moisture content. So this is going to allow seeds to experience moisture, experience temperature in their environment while excluding that moisture. Everything here is controlling for what happens in soil, so we can pick it apart and begin to try and replicate it in the lab and produce reliable germination protocols. We have seed that has been after ripened in the lab, and we have another replica here. Every month from April to September, retrieving these and plating them out at winter indicative temperatures that we've collected with data loggers across the range, or plating them out onto winter indicative temperatures and moving them to a medium to again test for warm stratification and cold stratification. This is what that looks like. There's 4 replicate sites in remnant Banksia woodland near Curtin University. This is what the monthly plate up looks like. We're doing about 600 to 700 plates a month. It takes about 5 to 6 hours to score. This is running in conjunction with a high density of data and temperature and moisture loggers sewn within the soil. So to explain this briefly, we have depth from soil surface along the top line, 0 centimeters, 1, 2, 3, 4, 5, so on. We've got 3 replicate sites, and that really shits me. 1, 2, 3. And we've got a time series along the bottom, starting in late January and going till the beginning of April when I last retrieved these. It's still running. What we see is a pretty extreme gradient in temperature. The Rutaceae, we know by their mass in loose Banksia soils, are going to move down the soil profile. And we expect that the ranges of temperatures that they're going to be exposed to are about here. So delving into this data with soil moisture, rainfall, and seed moisture content data that we've also been collecting, we're going to be able to analyze this with any germination that we get in the field and produce more informed methodology to test germination in the Rutaceae. I pulled up a retrieval in May, and I'm happy to say that these things are actually germinating in some cases. So Crowea angustifolia and Phylothecus baccata are 2 of the really problematic ones that we haven't been able to push for a while. I literally did this the night before I got on the plane, so I haven't had a chance to analyze the data. But in a replicate of 25, we're looking at maybe 8 to 9 germinants in this case. In a replicate of 25, we're looking at about 5 to 6 in the case of Phylotheca. So something is happening, and I'm hoping with a lot more work and a lot more investigation over the coming months, I'm going to be able to pin down exactly what's going on and hopefully inform a lab-based, nursery-based, and restoration-based practice to hopefully get some of these species and potentially others into restoration sites. Thank you.
Hello, Michael.
G'day,
My
mate.
name's Steve. that was great. Do you sort of look at country when you go out there? I mean, it's one of the things that I think that we do really well. It is have that sort of knowledge of the land, and sometimes you can just pick something up out of where the collections are coming from, or you know, do you do that?
Yeah, so it's been a while since I've actually been able to get out there and collect seed and actually get out into the bush. You do see a lot of things like that, mainly around timing of germination in the field. If you're able to identify seedlings, flushes of seeds coming up either after fire in one case or after fire breaks have been put in. That disturbance when the season changes allows for germination. I suspect that's either releasing soil-based carotenoids and smoke back into the system, or that removal of overlying vegetation is actually changing the diurnal temperature flux that you get, and that change in temperature is driving dormancy break. Lots of indicators, but to actually test it in a scientific way is a lot more. I get a lot of people, nurserymen, saying the Ericaceae was the last group I worked at, big woody fruits. People taking emu scats, processing them in the nursery, getting great germination, but to actually pick what out of the millions of factors go into that is quite difficult. But in a nursery setting, that's definitely what I would be looking at.
I sort of answered the question. I was just going to ask, do you see much regeneration of these species in the field, and is it episodic?
It definitely seems to be linked to fire, but I don't think that's the whole period. I think we do get into fire recruitment. I think we know generally what's driving these systems, but not specifically in these species cases, yeah. Cool. All right, could all join me in thanking me?